My name is Michael Zheng. I'm currently an undergraduate summer research intern under the supervision of Dr. P.S. Lala from the Department of Anatomy and Cell Biology. I'll be discussing the progress achieved during my undergraduate thesis project, which is titled Constructing an In Vitro 3D Model of the Human Placenta. Much of this project involved the contributions of Sarah Bhatia, my thesis partner, and was conducted under the guidance of my lab mentor, Zheng. The placenta is a temporary invasive organ of fetal maternal origin that supports the fetus during pregnancy. It facilitates the exchange of key molecules between the maternal and fetal circularization systems. The main fetal constituents are trophoblasts. Human trophoblast stem cells differentiate into two subpopulations. Syncytial trophoblasts provide exchange of endocrine functions. Extravillous trophoblasts invade the uterus and remodel the uterine spiral arteries to become wider, which increases maternal blood flow to nourish the fetus with oxygen and nutrients. The uterine endometrium also undergoes remodeling to form the decidua, which involves the differentiation of endometrial stroma cells into decidual cells. While the placenta plays a critical role in determining pregnancy outcomes, investigating human placentas through non-invasive methods is not feasible. Thus, there is growing interest in developing comprehensive in vitro models of the placenta for in-depth studies. While researchers have recently developed promising 3D trophoblasts and decidualized endometrial organoids, they do not adequately represent fetal and maternal components as 3D structures that interact with one another. Our project aims to fill this gap, which is demonstrated by our two objectives. We plan to distinguish placental cell lines by using cell-specific markers and co-cultures that represent the fetal and maternal aspects of placenta. Our second objective was to promote cellular interactions between trophoblasts and endometrial stromal cells by opposing fetal and maternal spheroids. We expected that our in vitro placenta models will only partially recapitulate the morphology of the placenta in situ, but that it can be improved upon further. We took the following approach to address our first objective. For our fetal components, we selected HCR cells, or HCRs, to represent the trophoblasts. To incorporate the fetal vasculature, we opted for fetally derived human umbilical vein endothelial cells, or HUVECs. And we have fluorescent probes that have been developed to track cells. So we incubated our HCRs with cell tracker green dye. We mixed these fluorescent HCRs with HUVECs in a 1-to-1 ratio to form fetal co-cultures. CD31 is an endothelial cell marker and should be present in our HUVECs. These fetal co-cultures were immunostained for CD31 and examined under a fluorescence microscope. For our maternal component, we designated telomere immortalized human endometrial stromal cells, or THUs, for the endometrial stroma. To represent the maternal vasculature, we chose to use human uterine microvascular endothelial cells, or HUMECs. We mixed the THUs with the HUMECs in a 1-to-1 ratio. And to mark the THUs, CD10 was chosen as a endometrial stroma marker, and CD31 was expected to be present in the HUMECs. We performed dual immunostaining for CD10 and CD31 with the maternal co-cultures and analyzed them under a fluorescence microscope. For our second objective, involving fetal maternal spheroids, we ultimately decided to omit the endothelium due to resource limitations. HCRs containing cell tracker dye were plated onto an ultra-low attachment plate, which was then centrifuged to form a spheroid by cell aggregation and allowed to grow for another 48 hours. We also created spheroids from THUs with a similar protocol. To catalog our process, we analyzed the HCR and THU spheroids with deconvolution microscopes at the Biotron facility. We applied Z-stacks to separate these 3D structures to 2D planes and applied an algorithm to correct for scattering play. We then plated spheroids with HCRs being cell tracker dye and THUs onto chamber sides. and we attempted to minimize the distance between them by using a pipette tip to manipulate their placement. For our initial attempt, the spheroids were examined under a time-lapse microscope that maintained culture conditions for viable growth. This process occurred for 24 hours. Phosphorescence and phase contrast images taken hourly emerged. Many of our results tended to be preliminary. This figure shows fetal co-cultures of HTRs and HUVECs. Cell tracker green tie was detected in these cells, and when detecting CD31, we found a distinct group of cells. In data not shown, the emission of cell tracker green dye or CD31 antibodies led to severely diminished green and red fluorescence in our controls. From these findings, we determined detecting cell tracker dye and immune stain for CD31 visualize distinct populations in fetal co-cultures that represent HTRs and HUVECs. When examining our maternal co-cultures, our dual immune stain for CD10 and CD31 to identify T-Hus and HUMECs was inconclusive. However, this figure shows that CD10 was sufficiently present T-Hus, reaffirming their endometrial stroma origin. We also visualized our spheroids with cell-specific markers under a deconvolution microscope. For the HCR spheroids, cell tracker green tie allows for characterization of spheroid shape and size, and likewise immunostaining for CD10 allow for visualization of T spheroids. At this point, we oppose the HCR and T spheroids and follow their progress under the time-lapse microscope. Unfortunately, the HCR and T spheroids remain physically separated, with drifting over time, and overall, meaningful cellular interactions between HCR and T spheroids were not observed, which could be attributed to inadequate juxtaposition at the onset. Interestingly, cell migration or sprouting from the HCR spheroid suggests EVT outgrowths that could be directed towards T-Hus in the future. From these results, we had two main conclusions. Detecting cell-specific markers distinguished cell lines in fetal co-cultures, and our initial attempt of opposing HCR and T-Hus spheroids was unable to replicate trophoblast invasion of the endometrial stroma. To the best of my knowledge, no other study has attempted to have spheroids of different types interact with one another. Overall, our findings laid a fundamental groundwork for developing an in vitro placenta model that better represents fetal and maternal components in 3D forms. While these techniques have potential, some next steps are needed to create a more comprehensive 3D in vitro placenta model. This includes establishing physical contact between HCR and T spheroids at the onset and following their progress over an extended period. A strategy for distinguishing between T-Hus and HUMECs in maternal cultures requires further investigation. Endothelial elements like HUVECs and HUMECs should be incorporated into the fetal and maternal spheroids for added complexity. And eventually, we aim to create in vitro 3D models that simulate pathological placentas for novel therapeutic insights, which could improve health outcomes for both mothers and fetuses. To achieve this objective, we plan to alter the expression of decrin, a proteoglycan produced by decidual cells in T-Hus. Our lab found that decrin restrains trophoblast invasiveness, and moreover, its overexpression in decidual cells is associated with preeclampsia in mothers with or without fetal growth restriction. Both preeclampsia and fetal growth restriction are conditions that involve a hypoinvasive placenta. It has also been postulated that a decrin deficiency in the decidua may lead to a hyperinvasive placenta, also known as placenta accreta. I hope to address these inquiries throughout my internship and upcoming master studies. This research was funded by grants awarded to Dr. P.S. Lala from CIHR and NSERC. I'd also like to acknowledge the gracious assistance provided by members of Renault Laboratory, Carrie Nygaard of the Biotron facility, and Dr. Lin Zhao. Thank you for listening to my presentation, and I look forward to answering any questions you may have in the chat.